Hello, I am Kanchan Kumar, a final year student of B.Tech Civil Engineering at Faculty of Engineering and Technology, University of Lucknow. Today, I am going to give a seminar presentation on dual-chambered microbial fuel cell to treat wastewater and electricity generation. So, let's get started. In this presentation, we will discuss about what is microbial fuel cell, its working principle, types of the MFCs, typical hierarchical architecture of MFCs, components, working of the MFCs, parameters used for evaluating the MFC performance, microbes used in the MFC, applications of the MFCs, case studies, advantages, limitations of the MFCs, and finally conclusions. Introduction. Currently, the world is facing the problem of power scarcity, pollution from fossil fuels, and the problem of selecting the appropriate technique in sewage treatment. Renewable and clean forms of the energy are one of the society's greatest needs. Use of the fossil fuels has contributed to global climatic changes, environmental degradation, and health problems. Microbial fuel cells have potential to treat wastewater and to generate electricity as well. Microbial fuel cell technology offers the possibility of harvesting electricity from organic waste and renewable biomasses. MC Potter was the first to perform experiment on microbial fuel cells. What is MFC? A microbial fuel cell, MFC, is a device that converts chemical energy to electrical energy by the action of microorganisms. Microorganisms are capable of converting a wide variety of biodegradable organic materials into carbon dioxide, hydronium ion, and electrons. Basically, MFC contains a anodic chamber, a cathodic chamber, a proton exchange membrane, and an electrical circuit. We will discuss about them on later slides. Principle. The working of microbial fuel cell is based on the principle of redox reactions. Bacteria oxidize the organic matter to produce carbon dioxide, electrons, and protons. The substrates are converted into electrons by bacteria. Electrons run through the circuit to generate power. Types of MFCs. Basically, there are two types of the MFCs. The first one is mediator-free MFC and second one is mediator MFC. So let us discuss about mediator MFC. If the microorganisms or organic matter consumption capacity is very less, then some enzymes or chemicals are added in water to increase the efficiency of the MFC. Then that type of the MFC is called mediator MFC. Now we will discuss about mediator-free MFC. In this, if the microorganisms present in the wastewater is capable of generating sufficient amount of the power by consuming the organic waste present in the wastewater, then there is no need of the mediator in MFC, and that type of the MFC is called mediator-free MFC. Typical hierarchical architecture of MFC. Basically, MFC consists of a anodic chamber, a cathodic chamber, a proton exchange membrane, and external circuit. The anodic chamber consists of anode, substrate, microorganisms. Cathodic chamber consists of cathode, reducer like oxygen. The example of the proton exchange membrane can be an FEON. Now, the anode is made up of the carbon rod, graphite rod, or can be in a powder form as well. Similarly, the cathode can be made up of cathode rod, graphite rod, powder form, etc. Now we will discuss about the components of MFCs. The first one is anode. Anode is conductive in nature. It is biocompatible and chemically stable with substrate. Anode can be made up of the graphite and can be in the form of the plates or rods. Bacteria live in the anode compartment and convert substrate to carbon dioxide, protons and electrons. The second one is cathode. Electrons and protons recombine at the cathode. Oxygen is reduced to water in the cathodic chamber. The cathode can be made up of the graphite and can be in the form of the plates or rods. The next one is exchange membrane. Exchange membrane is a thin membrane which is placed in between the anodic and the cathodic chamber. Now, the protons from the anodic chamber is transferred to the cathodic chamber by the exchange membrane. The example of the exchange membrane can be a Nafion or Ultrex. Protons and electrons recombine on the other side, that is on the cathodic chamber. The next one is the electrical circuit. After leaving the anode, electrons travel through the circuit. These electrons power the load. The next one is microbes. Basically, the microbes is an extremely small living thing that we can only see with the help of the microscope. An anaerobic microorganism or anaerobe is any organism that does not require oxygen for growth. The examples of the microbes can be Actinobacillus cyanogenus. The next one is Eramonas hydrophilia, Irvina dissolvent, and many more. The next component is substrate. The substrates provide energy for the bacteria cell. Waste that contain the organic substrate like carbohydrate, protein, cellulose, etc. Now we will discuss about the working mechanism of MFCs. The wastewater is filled in anode chamber. The microorganisms present in wastewater are going to consume organic waste and converts it into carbon dioxide, H plus ion concentration and electrons. Now these electrons flow through the wires. The circuit consists of a resistor, a multimeter, and the complete circuit is connected to a computer. With the help of the various softwares, we can calculate the amount of the electricity that had generated. Now, the H plus ions are going to flow from anode chamber to cathode chamber through proton exchange membrane. Proton exchange membrane allows only H plus ions to pass. That means only protons are going to pass through anode to cathode. Other particles are obstructed by proton exchange membrane. Air disperser can be provided in cathodic chamber to provide fresh molecule of oxygen. Molecules of oxygen and hydrogen which had passed through the proton exchange membrane can combine together to produce the fresh molecule of water. So, in this way, the wastewater is treated and the generation of the electricity takes place as well. Let us see some parameters used for evaluating the MFC performance. Loading rate. Its unit is kg per meter cube day. It is an amount of the organics in kgs loaded to MFC. Effluent quality. Its unit is kg per meter cube. It is the concentration of the organics in an effluent discharged from the anode chamber. Treatment efficiency. It can be expressed in the percentage, and this can also be referred as COD removal efficiency, that is estimated by dividing the COD concentration in the effluent by that in influent. Current density. Its unit is ampere per meter square. It is considered to be an index related to the total catabolic activity of the microbes in the anode chamber. The current generated is normalized to an anode area. Closed circuit voltage. Unit is V. It is the voltage between the anode and cathode measured in the presence of the resistance. Energy efficiency, and it can be expressed in the percentage. This is how much power we are expecting and how much power is generated. Now the last one is Coulombic efficiency, and this can also be referred in the percentage. This is defined as a ratio of the coulombs measured as the current to the total coulombs contained in the substrates. Various microbes used in the MFCs. The first one is Actinobacillus subnogenus. Its substrate is glucose. If we are using this microbe, then we have to use a thionin as an electron mediator. The second one is Eramonas hydrophilia. Its substrate is acetate. And if we are using this microbe or this type of the microbe is present in the wastewater, then we have to construct a mediatorless MFC. The second one is the Erwina's dissolvent. Its substrate is glucose and its application is ferric chelate complex as a mediator. We have to construct if these type of the microbes are present, and many more. Now we will see the applications of MFCs. MFCs can be used for power generation. It can be used for wastewater and sewage treatment. MFCs can be used in the biosensors and biogas. Now we will discuss about the first case study. This case study is based on generation of the electricity from dye industry wastewater in dual-chambered microbial fuel cell. Initial characteristics. The dye wastewater was collected from Tripur district, Tamil Nadu, India. The wastewater is stored at 4 degrees Celsius. The physical chemical properties of the dye wastewater, such as pH, total suspended solids, total dissolved solids, total chemical oxygen demand, soluble chemical oxygen demand, was shown in the table, and the various range of the parameters are given below. Microbial fuel cell construction. Dual-chambered microbial fuel cell consists of two chambers, anode and cathode, which is made up of plexiglass. Each of the chamber had a height of 11.2 cm and diameter of 7 cm. Anode and cathode are interconnected with a flange gasket which holds the proton exchange membrane Nafion 117. The projected surface area of an activated carbon fiber belt was 169 cm square. The two electrodes are separated and placed at a distance of 5 cm, which is connected to an external circuit using insulated copper wires with a resistor. The measurement of voltage and current is done through a digital multimeter. MFC operation. Total chemical oxygen demand of dye wastewater was diluted in the range of 800 mg per liter and then introduced into the anodic compartment. Inoculum used in this study is taken from the secondary clarifier of the municipal wastewater treatment plant. To prevent the growth of the methogens, 2 mole per meter cube of the bromoethene sulfonate is added to the anode chamber. The experiment was operated in different hydraulic retention time from 35 days, 30 days, 25 days, 20 days, 15 days, 10 days, and 7 days. In every hydraulic retention time, chemical oxygen demand removal efficiency, solid removal efficiency, and power generation were verified. Results: the efficiency of the total chemical oxygen demand removal of 66.10%, 29.58%, and 52.59% were obtained at the hydraulic retention time of 35 days, 20 days, and 7 days respectively. The suspended solid removal efficiency was 63.43% and 41.27% at the hydraulic retention time of 35 days and 7 days respectively. At the hydraulic retention time of 35 days, the voltage was 235 milliwatt. At that period, power density is 8.94 milliwatt per meter square was obtained. The highest power density of 96.85 milliwatt per meter square was achieved as the hydraulic retention time of 20 days with the voltage of 649 milliwatt. The Coulombic efficiency at 35th day, 20th day, and 7th day was found to be 21.61%, 60.54%, 17.56% respectively. Conclusions: the dual-chambered microbial fuel cell operated to treat the dye industry wastewater accomplished the generation of the electricity along with the removal of the organic matter. The maximum power density of 96.85 milliwatt per meter square was obtained at a hydraulic retention time of 20 days with the Coulombic efficiency of 60.54%. The maximum TCOD and SCOD removal of 66.1% and 57.94% respectively were achieved at the 35th day. The MFC could be a suitable alternative for the removal of the biodegradable components present in the dye industry wastewater with the power generation. Let us see the second case study. The second case study is wastewater treatment with microbial fuel cell, a design and feasibility study for scale-up in microbreweries. Design data of MFC. The wastewater was collected from a craft brewery located in Ontario, Canada. The MFC, using graphite brush anodes, cloth cathodes, and a Nafion N115 membrane, was constructed. The final design consists of two MFCs system in series. Each MFC unit has an anode and cathode chamber separated by a semi-permeable membrane of Nafion N115. A surge tank was constructed to accommodate the difference between wastewater production and treatment capacity. Two such tanks, each of capacity 1000 liters, was used. Pumps are capable of processing 105 liters per hour of wastewater. Brewery wastewater was fed to the first anode chamber, where bacteria oxidize the organic material, producing a stream lower in organic material, which can be fed into the second MFC in series. Water leaving the second anode has a composition lower than the municipal discharge requirements making it suitable for discharge directly down a sanitary sewer. Results: the wastewater generation rate from brewery industry was found to be 256 liters per hour, and MFC can treat wastewater with a rate of 84 liters per hour. Initial COD was 3000 mg per liter, and the final COD after treatment is found to be 243.67 mg per liter, which is representing a COD conversion rate of 91.88%. Electricity generation is 26.4 kilowatt-hour. The capital cost is $289,000 and net present value is $113,000. The payback period is 5 years. Based on payback period and net present value, the scale-up of microbial fuel cell appears to be feasible. Let us discuss about advantages and limitations of MFCs. Advantages: generation of the electricity from bio-waste or organic matter. MFC is an alternative source of fuel. Bioremediation of toxic compounds, wastewater treatment and power generation at the same time. It is eco-friendly and can be applied in area lacking electricity. Limitations: low power density, ohmic losses, required high level of maintenance and functionality, high operational and maintenance cost. Now, let us see the conclusions of MFCs. Microbial fuel cell technology is clean and effective technology. MFCs have been explored as a new source of the electricity generation during operational wastewater treatment. It can be used for the production of the secondary fuel as well as in bioremediation of toxic compounds. However, this technology is only in research stage and more research is required before domestic MFCs can be made available for commercialization. These are some references. Thank you.